Hello, we're glad you've joined us for this live webinar, Pediatric Genetics, Applications of NGS in the Clinic. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Let's get started. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, and to get your credits, click on the button located in the bottom left-hand corner of your web page. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, EmilyFarrow, PhD, Director of Laboratory Operations at the Center for Pediatric Genomic Medicine at Children's Mercy Hospital. Dr. Farrow joined the center as a research scientist in February 2012. She is an assistant professor at the University of Missouri-Kansas City School of Medicine and has a secondary appointment at the Indiana University School of Medicine in the Department of Medical and Molecular Genetics. Dr. Farrow obtained a master's in genetic counseling from the Indiana University School of Medicine, followed by a PhD in molecular genetics. She has completed a postdoctoral fellowship supported by a National Kidney Foundation research grant in phosphate regulation at Indiana University School of Medicine. Dr. Farrow is also a board-certified genetic counselor. Her research interests include metabolic bone disorders and the role of genetic counseling in genomic medicine. Dr. Farrow will now begin her presentation.
Thank you very much for asking me to give this presentation. I'm happy to share a little bit about our center and how we're using genomic sequencing. Let me bring up my slides. So again, I'm going to talk about pediatric genetics and specifically how we're applying this in our clinic and some of our research applications. So a little bit of background about our center. The Center for Pediatric Genomic Medicine at Children's Mercy is a relatively new center, and we were established in January of 2011. And at the time, we were one of the first pediatric genomic centers fully immersed within a pediatric hospital. Our center is rapidly enlarging, as you might expect, and we're made up of a large team and essential people of directors such as myself over the laboratory, medical directors, bioinformatics. We have research coordinators, and we have an entire informatics team that supports us that I'll talk about. We have physicians that we work with throughout the hospital, and then also, as you might expect, we have essential collaborations with our Center for Bioethics and our clinical genetics program. And really, it takes a whole village to run a genome center. So one of the things I want to spend a few minutes on in the presentation is just giving some context about the human genome and next generation sequencing in general, because I think if you have a basic foundation, it makes it much easier to understand different types of testing and how they're being applied. Otherwise, some of the words can quickly become Greek and not make much sense. So to keep it in perspective, the human genome is 3.2 billion base pairs, or that's 6.4 billion nucleotides. And if you're going to copy that yourself manually, that would be equal to typing 60 words a minute a day, 8 hours a day for 50 years. So when you think about what our bodies are able to do, it's amazing. Now, of those 3.2 billion base pairs, there's only about 19,000 genes. So this is 1 to 2% of the genome. So we're still working on understanding what the rest of the genome, the other 98%, is doing. But those 19,000 genes are coding at least 100,000 proteins. And these numbers change a little bit, I feel almost monthly, whether there's more or less genes. But this is a pretty good number that's been fairly stable. And so one of the things to think about too is the relationship of frequency and size and the types of genomic variants. And this also helps when you're looking at testing that you might order in the clinic. So this chart is just showing you that structural changes happen least often. So this is where we have chromosomal rearrangements, duplications, or trisomies, but they cover the biggest space because they are so big. Then if you move to the next smaller, we have copy number variants, and this is where really we've seen microarray be especially informative. So this is where we're looking at big gains or losses within a chromosome. We then are going to a little bit smaller insertions and deletions, so there can be SNP arrays. There's also recently been the development of exon arrays, and these will give you deletion and duplication information on an exon by exon basis within genes. And then for genomic sequencing, really what we're focusing on here are some of the smaller variants, so single nucleotide variants. And while they are the smallest variants, if you look at the number of nucleotides, they happen the most frequently throughout the genome. And this has been the bread and butter of genomic sequencing and really will continue to be for the near future. And so this is one of our favorite quotes or descriptions when we think about next generation sequencing and how that's being a part of personalized medicine. And genomic medicine is really a new structured approach to disease discovery, diagnosis, and management that's prominently featuring these next generation sequencing techniques and analysis. And the title of this was Charting a Course for Genomic Medicine from Base Pairs to Bedside. So I think we're really now at the cusp and we're really realizing how genomic medicine is changing our clinical management, and it's just an exciting time. So traditionally, when you think of genetics and the way that it's been done in the past is that we had Sanger PCR, and that's where we were doing gene by gene, exon by exon sequencing. And so what would happen is that a patient would come into clinic and we would look at them and we do a very detailed phenotypic assessment to say what clinical features does this patient have, and then I think this is most representative of this disease. So an example might be a patient that comes in with multiple fractures. We say, gosh, they really look like they might have osteogenesis imperfecta. So we're going to order collagen 1A1 screening, and that's a big gene, so it would take a while. Now, in some cases, you can do multiplexing with Sanger PCR, and that accommodates small panels, but these are still time-consuming, and they're really limited to known genes, so you have to know where to look in order to do this type of sequencing. The plus side, and why this is still called the gold standard in many cases, is that Sanger PCR sequencing is very precise. So if we look at a patient and we know where to look for that change, we can see here that they're heterozygous for a change. And if we look in our control, our negative control, they're homozygous for the reference. And so it's very clear that there's a change, a heterozygous change in this person. But we're seeing a shift, and what next generation sequencing is that these are technologies that permit rapid interrogation of DNA up to and including entire genomes via massively parallel sequencing. And this is really a term that emphasizes the distinction from the initial approaches that involve sequencing of one DNA strand, one gene, one exon at a time. And because we've seen this rapid decrease in cost and a rapid decrease in time, it's creating a paradigm shift in the approach to monogenic disease particularly. And so I mentioned that our center was one of the first centers wholly immersed within a pediatric hospital, and I think there's lots of reasons of why next generation sequencing in genomic medicine is particularly applicable in children. And there's some hard facts to that, and that's because currently there are over 8,000 suspected genetic inherited diseases. But they're affecting 1 child in 30 in the United States. They cause 1 in 6 children's hospital admissions, and they actually can cause up to 20% of infant deaths. It's really significant when we're looking at our healthcare population. We look at those 8,000 suspected genes or diseases, we only know the molecular basis for about 4,700 of those. So we still have almost 3,000 gene or inherited syndromes where we don't know the cause or the underlying molecular diagnosis. And this means that a diagnosis can still often take years for a family, and I think we all can agree that a delayed diagnosis may delay appropriate treatment, it may allow a disease to worsen, and it can hurt a family in that not knowing the cause is very frustrating and can be hard. And it also can cost money, not that that's the important factor, but it's costing money and time and test ordered and clinic visits trying to figure out what the answer is. So this has been something that we've seen over and over, and I'll show with case examples, is that we're now seeing a shift. So part of this paradigm shift is that before we were really based on a phenotypic-driven diagnosis, and we're replacing that with a genotypic diagnosis. And next generation sequencing is what's enabling us to make the diagnoses of these atypical presentations that you maybe would have never considered otherwise, that gene or gene family. So I'm in genomics. I'm part of the center, obviously, as I'm running the lab. And still, to me, to this day, oftentimes it can be overwhelming, the number of sequencing options we have that can be used both in a research and clinical basis. And so what are the projects that we're doing, and how is sequencing being done? Because I think this cartoon summarizes it really well. It can be just overwhelming at the beginning. And so clinically, we have three main buckets that we're using for our testing that I want to go over and really highlight how next generation sequencing is being applied and changing our practices. So one is a targeted panel called TagScan that we developed. It includes 571 genes. These are mostly autosomal recessive pediatric onset genes. There are a few very common autosomal dominant, again, such as the collagen genes for OI. And this includes the mitochondrial genome as well. We also have exome sequencing that we're offering on both the research and on a clinical basis. And again, whole exome sequencing is going to cover all of the 19,000 known genes, and this is going to sequence every exon of those genes. We also supplement and include the mitochondrial genome. This has been really good for novel disease discovery and none of our medical mysteries. And then we also, as a center, offer clinical whole genome sequencing for our patients. And we also have some whole genome sequencing on a research basis, and you may have heard of our STAT-seq, which is our rapid protocol that I'll go over. And I think this is really highlighting the intersection of research and clinical and how that line is becoming grayer and blurrier all the time. So I want to spend a few minutes on how a next generation sequencing works, the technology, because it'll make much more sense to you then what the difference between a targeted panel, an exome, and a genome. And so just briefly, the sample preparation for next-gen, while there are subtle differences between the tests, they basically follow the same procedure. And one of the critical things is that all samples undergo library preparation. And really the goal of this library preparation is to add our sequencing primers, and also we add barcodes to identify individuals. So similar to a UPC symbol you might see in the grocery store, we're barcoding our samples so that we can know who that DNA belongs to, and also so that we can mix them downstream together. Another thing I want to point out that for short-read sequencing, this process involves fragmenting the DNA. And so this is where the term shotgun sequencing comes from, because we take the DNA and we break it up to a lot of pieces, just like we hit it with a shotgun and broke it up to thousands of pieces. And this is a picture of one of the instruments that we use in our laboratory to help with the library preparation. If we're doing a whole genome sequencing, all we have to do is library preparation, because we want to sequence the entire genome. So all we need to do is add our sequencing primers and our barcodes, and then we're ready for sequencing. So that's one of the main reasons why we've used Genome, and others have used genome sequencing for rapid sequencing, because even though you're sequencing more, so we're sequencing a whole genome, the sample preparation is actually very short. If you're doing anything less than that, so if you take the whole genome minus one gene only, which you wouldn't do, but as an example, you need to do something called enrichment. And this is where targeted panels and whole exome sequencing come in. And this really is involving selecting out specific regions of interest for sequencing. So those 571 genes, we're saying this is all we want to look at. And we use capture probes to grab the areas that we want to sequence. And then we discard the rest that we don't want to sequence. And we do these for multiple reasons, but some are cost and time considerations. So if we're sequencing less, then that means that we can, per sample, that means we can put more samples together. And so then that becomes more efficient from a time perspective. We also are able to pool samples together for our enrichment. So we take example of those unique barcodes, and that allows us to mix samples together so that we can have up to 12 exomes in a single tube for preparation, which makes it easier on the laboratory side. So this is decreasing the cost because we're doing samples together in one tube, and we could sequence more on a single instrument. But we also get a decreased time for the collection. So if you were just doing one sample, then a genome would be faster. But if you want to sequence 12 people at a time or 96 people at a time, then it's much faster to do the exome and move them together. Down below, what we're showing is really kind of just an example of how our enrichment strategies work, or how everybody's enrichment strategies work. So here's a depiction of an exome. The black bars are the exons that we want to sequence. And then up above, you see where we've tiled over those capture baits, so the areas that are complementary. And what they're going to do is they're going to grab those 3 exons that we want to sequence. And here we don't want to sequence this last one. So we don't have any baits. So that means when we go to wash and discard our supernatant, we'll get rid of that and we won't have to sequence that on the sequencer. So after our preparation, so whether we do library target or whether we do genome, targeted, panel, or exome, then we're ready for sequencing. And so within our center, we're fortunate to have 5 HiSeqs, ranging from HiSeq 2000 all the way up to a HiSeq 4000. And this enables us to have pretty broad sequencing capabilities in that we can do 1 genome in 25 hours on 1 instrument, all the way up to doing 16 genomes in 3 days on that 4000 on 1 single instrument. We also have 2 MiSeqs that we use for sequencing. These are smaller instruments. They use the same sequencing chemistry though. And so these allow us to do 4 tag scan samples. So again, that's that small panel and about 30 hours per MiSeq. So we could do 8 at a time if we run both instruments. So sample preparation and sequencing are really only one part and one small part of the process, although they're a critical one. But what we really have is a critical need for bioinformatics. And so I often feel like the person in this cartoon in that I'm really good at sample preparation, sequencing, and generating the raw data. And then a whole bunch of stuff happens on our computers that then give me the data to be able to do the analysis. So to be able to support that stuff, all of the compute that needs to happen, we have a large bioinformatics team that's developed a suite of custom software. And so one of the things that we need to do when we first get our sequencing or when the data first comes off of that sequencer is that we need to be able to take that raw data and turn it into variants that we can annotate. And so what I'm showing here, the white bar across the top, is our reference genome. And below that, those 100 base pair segments are representative of what a single read might be from a sequencing or a patient that we've sequenced. And so after the data comes off the sequencer and we do our calling to find out what our variants are, what we have to do is take that data, take those reads, and we align them back to the reference genome. And that allows us then to compare and say, yes, this person is wild type, they match the reference, or no, this person actually has a change, as you can see here, where they have a heterozygous change from the reference sequence. And so what this is allowing us to do then is identify all the places in the genome where our person is different. After that, after we identified all the places where we have a variant from the reference, what we then need to do is use our software to characterize so that we understand whether this is just a common change or is this a change that we want to look more into. Is it in a gene or is it in between a gene? And give us all that information that enables us to do the analysis. So one of the last things I wanted to talk about sequencing and what you'll hear referenced many times when you're looking at sequencing is sequencing coverage. And this is what's describing the average number of reads that align to or cover known reference bases. So in the previous slide, you saw that there are multiple reads that were tiling over a single base. And as you cover or read over a single position more times, so the base calls can be made with a higher degree of confidence. So it's the same thing as if you read this slide 5 times, you would have a better feeling or feel more confident that you knew what was on the slide as if you'd only glanced over it once. This is particularly important as we think about things that are outside the box of inherited Mendelian disease, such as cancer, where we have somatic changes, where you might have a cancer clone that's only there, say, in 5% of the DNA. And so you need to read over that same position multiple times so that you can catch the changes that are in that lower level. It's also important because in contrast to Sanger sequencing, NextGen still has inherent error rates. And that means the probability that at any given base is the right call. And what's a score that we use is called a quality score of Q30. And what that's saying to us is that there's only a 1 in 1,000 chance the nucleotide call is incorrect. So while most of us would be fairly confident and would bet on those odds, it's still better practice to read over multiple times so that you have a higher confidence in making that base call. And so at the end of the day, if a gene is covered on genome, on the genome, which it would be, covered on exome and on tag scan, the difference between them then is really your coverage. And so targeted panels are allowing for deeper sequencing, so you're going to read over that base more times with less overall data. So standard coverage for a genome would be 30x, and that really tends to be the minimum that most people use. You're going to read over every base 30 times. An exome on average, you're going to read over the bases that you want to sequence 80 times. And when we get down to our targeted panel, only 571 genes, we're going to read over it over 200 times. So what does this look like in real data? So this is just a picture of the cystic fibrosis gene, CFTR, which is one of the most common inherited diseases in North America in the US. And so here we're just showing you all the horizontal purple lines here are the exons of the gene. It's a fairly large gene. And so when we think of that, those are all the exons that we're going to want to cover when we're sequencing. And so here I've showed you representative coverage over the same exact area in both a tag scan, an exome, and a genome. So you can see on the bottom in the genome that we have nice even coverage completely across that region. But you will see a small little gap in the coverage, and that's because even with genome sequencing, there are areas that are very repetitive that are difficult for the technology to go through. You can see then in the exome and tag scan that we have these humps of coverage over the exons. And so these are areas that we've wanted to enrich and that we have nice deep coverage on. You can see in tag scan, which was a second hump that's not in the exome though. And this is an area that is there's an intronic change that we know is pathogenic. And so because we custom designed this panel, we're able to go in and say, hey, we need to get this area too. This exome data that I'm showing you is just an exome off the shelf, so it's designed to cover all the genes, so it didn't have that intronic step to cover. So when we zoom in closer, we can see over the exact same position. When I say coverage changes, we read over the same position in that tag scan over 200 times. We read over it in the exome about 72 times, and we read over that same position about 30 times in the genome. So again, it's how many times. Coverage is how many times did you go over that same region to gain confidence in that call. That's one of the main differences in the different types of testing. So as you might imagine, if you're looking at different numbers of variants or different numbers of genes, you're going to get very different numbers of variants to look at. An analysis of next generation sequencing data has been described as finding a needle and a stack of needles. It's one of my favorite quotes. And so from tag scan and 571 genes, what we get is on average 8,000 variants for us to look through and analyze. An exome can be anywhere up to 300,000 variants, and a whole genome can produce up to 4 million variants. None of us have the time to sit and look through even 8,000 variants, let alone 4 million variants. So again, this is where software is going to come in and tell us and help us narrow down which variants are just normal population changes and which variants are rare and in genes that we need to look at and might be potentially pathogenic. So this is again where our software suite that our center has developed comes into play. And so this is for our variant annotation, classification, and analysis. And I'll go through what these are doing and show how this works. But RUNES is our first one. It's for variant annotation. SAGA is mapping our clinical symptoms to genes. So that's giving us a short list of genes for us to look at. And then VIKING is that final piece that's going to integrate all of our data. So our characterized variants with our prioritized gene list and allow us to look through it in real time. So RUNES, we like acronyms in our center. It's the Rapid Understanding of Nucleotide Variant Effects software. And for every single variant that we see, what RUNES is doing is it's going through and giving us information about that variant. So it's telling us how frequent have we seen it or how novel is it. Have we seen it in 2,000 of the samples we've sequenced internally, or have we seen it only once? It goes out and looks at public databases such as dbSNP. We look at Exact and also 1,000 Genomes. We also pull in things like HGMD. Another thing that we've looked at and that is very important or helpful for us in our analysis is our ACMG categorization. And so this categorization is one that's been put forth by the American College of Medical Genetics, and it helps us to take all of our variants and put them in large buckets, which makes it easier for analysis. So category 1, for example, is a variant that's been previously reported and recognized the cause of disorder. So an example might be delta F508 and cystic fibrosis. We've seen that variant, and we know that even though it's common, it's associated with classic CF. Category 2 are novel variants, so they haven't been seen before, but they're the type expected to cause the disorder. So these are things like a premature stop codon, a loss of the initiations affecting the start, or frame shifts, or non-CIPS variants. So it's easy to interpret how those changes in that gene, or how the changes are affecting the gene. Category 3 are novel, so they're not very frequent, but it's hard, they may or may not be causative. So this is where our missense variants are coming into play, our in-frame insertions or deletions. And then category 4s are really novel, but probably not causative, so these are probably population changes. You can imagine that as each bucket, so category 1 is the smallest bucket, category 2 gets bigger, category 3, there are more variants that fall into that. So as you increase in category, you increase in variant numbers. So I mentioned that we're able to use SAGA, which is symptom and sign-assisted genome analysis, as a way to take the phenotype that's described from the patient from the clinic and turn that into a gene list to prioritize for our analysis. And this is a test ordering interface that it integrates with the electronic medical record. And again, it's mapping those clinical features to genetic diseases to genes. And it's serving multiple functions, but 3 of them, as I mentioned, are to nominate a superset of genes for for And then because most of our analysis is in the pediatric population, we have to think about incidental findings and guidelines for testing in children. And so this is a way for us to restrict our analyses to only genes that are associated or are known to be associated with that phenotype, and not look at genes that have absolutely no connection. It also is helping us, again, take those 8,000, those 300,000 variants and get a smaller list that's more manageable to go through for analysis. And so again, because most of our testing is in children, also from my genetic counseling standpoint, we think about how we're doing our testing and how it impacts our patients. And traditionally, if you look at genetic testing, there was previously a mandate to avoid violating a child's right to an open future. So as an adult, I may want to know that I have an increased risk of Alzheimer's disease so that I can be screened more often, or I might make changes in my diet or exercise, whatever I might feel is necessary. But a child may not want to know that. And if we told them that information as a child, if they didn't want to know that, they can't unknow the information. So really, we've tried to not tell children genetic testing results that's not impacting their immediate care, and allow them to make that decision when they reach adulthood. So this has impacted things like our test design, hence SAGA. It also helps us, has impacted how we approach testing and whether we need consent for testing and how we're giving back those results to the communication. Some of the things that we use to help us, again, is that's where clinical panels can be useful, is that you're only looking at disease-associated genes or genes that are associated with that phenotype, so such as tag scan, which is 571 genes. Or our exomes we offer as symptom-driven exomes, meaning that we're only going to look at the genes that are known to be associated with their features. Again, the last piece of the puzzle is our VIKING, so variant integration and knowledge, interpretation, and genomes. And this, again, is a way for us to integrate that SAGA and RUNES data. It also allows us, again, to prioritize those variants. And importantly, it's allowing us to filter on the frequency of the variant. So we keep track of that both internally in our own internal warehouse, and also we use, again, access public databases such as EXAC, and as well as inheritance filtering. So now I think the best way to understand how this information is being used in clinic is to use some case examples. And so this is a case example of tag scan, our panel. So our very first case that was referred to the center when we first started were CMH1 and 2. And these were sisters with progressive ataxia. So at the time they were sent to the Genome Center for sequencing, the first sibling, the older sibling, was 9 years old, and the second sister was 5. The first sibling had normal delay. She did have disordered movements and had ataxia that onset about the age of 4 to 5 years, which had resulted in her being wheelchair reliant by 8 years of age. And by 6 years of age, they had noticed that she had a progressive cerebellar atrophy. Now, the Younger sister, again, had mild delays in early development. Her ataxia onset at 2 years. It's not clear entirely if she's more severely affected, or it's just that once you have one affected child, then you're more attuned to symptoms that may have been in the first child. You just weren't looking for them. But she also was noted to have progressive cerebellar atrophy. This is an MRI from one of the children. And so I am not a neurologist myself, but I think it's very clear that there has been some, if you look at the MRIs, that you can see the cerebellar atrophy in these children is quite striking. So before they had gotten to the Genome Center for sequencing, this family had had 5 years since that first initial diagnosis with no diagnoses, and we had spent over $23,000 in testing, and this really included the gamut of all clinically very appropriate testing, from a karyotype to microarray to specific single gene testing for something such as ataxia, telangiectasia, down to some biochemical findings to try to see if we could get a biochemical diagnosis for the family. So we like to say, or we describe this family as being at a diagnostic crossroads when they got to the center. So this is how SAGA is working. Again, SAGA is being applied to tag scans, so 571 genes. So if we put in the term ataxia, out of those 571 genes, we've now come down to 78 genes. If we put in a second symptom from this family of a developmental disorder of motor function, we have 44 genes. You can see there's some overlap in the genes between ataxia and developmental disorder of motor function. And if we put in a third feature in SAGA, this is nominating 28 genes, and again, there's some overlaps with muscle atrophy. And if we look at the intersection of all three of these, we actually come down to a single gene of aptx. So when we did the sequencing, the family did actually have, were found to have a homozygous, the sisters, mutation in aptx. And this mutation stops the aprataxin protein from being made, and the parents, there was known consanguinity in this family, were found to be carriers of the mutation. And so we immediately, before we even ran the test, we were able to use SAGA to get down to one single candidate gene to start with. And that's where, in this family, we were able to find the mutation. Now, of course, they're not always this clean, but I think this is a very good illustration of how we're able to use software to, with our analysis. The same things can be applied to exome sequencing. And so we initially offered exome sequencing only on a research basis within our center. And so one of the things that we did before we turned it on clinically is that we looked at the clinical utility of next-gen sequencing in neurodevelopmental disorders. And we took the first 100 families that were enrolled in our research biorepository, which included 119 patients. And this was actually published almost 18 months ago in Science Translational Medicine by Soden et al. But what we found in looking at those first 100 families is that 45% of the children that had been followed in ambulatory care received a diagnosis after exome or genome sequencing. And the cost of the testing prior to any of the next-gen tests was $19,100 per family. And this suggested that exome or genome sequencing would be cost-effective at up to $7,600 per family, which is the cost that's easily attainable for exomes in the current state. Of those families, a change in the clinical care impression of the pathophysiology was reported in half of those diagnosed families. That's huge. So in half of our cases that got a diagnosis, their clinical care was impacted. And had we performed exome or genome sequencing and got a diagnosis at the onset of symptoms, we may have made a diagnosis on average 77 months earlier than it occurred in the study. That's almost 6 years. Which again, for a family, think about the journey they had been on with no answer. That's critically important for them. Another thing that we looked at is what are the inheritance patterns that we're seeing, and this is a neurodevelopmental disorder, and we had a wide range of diagnoses, but we were somewhat surprised at the time that de novo dominant were the most common. And for our studies, for our genome and exomes, we try to sequence the trio, meaning the affected patient and the parents when they're available, as often as possible. And that's giving us a strength to find those de novo variants that you wouldn't be able to categorize without the parents. And this is broadly applicable, though, well beyond neurodevelopmental. So we're seeing this more and more, that de novo dominant disease is much more common than we initially thought. After that, we saw things that you would predict, such as heterozygous autosomal recessive disease, dominant disease, and then mitochondrial. So again, I think a case can tell you and easily or illustrate how this has changed or isn't directly impacting our patients. So case 2 was a 13-year-old male who had fallen at home and complained of back pain. And subsequently, he developed numbness and tingling and was presented to the emergency room. And x-rays revealed multiple compression fractures of the spine, which is not something that you would see in a child typically who just falls. His bone density score revealed severe osteoporosis with a z-score of -4.7, so that's quite significant. And the patient had reported a history of 3 arm fractures previously with appropriate trauma, so he'd fallen just doing normal boy things. But there was no report of low bone mineral content or osteopenia on any of his prior scans. So he was sent for evaluation, and collagen 1A1 and 1A2 gene sequencing were ordered appropriately for osteogenesis imperfecta. And of course, the results came back with a variant of unknown significance. So this is an intronic variant that hadn't been seen before. And so they hypothesized that it might be affecting splicing. So parental testing for the variant, so they tested both parents for this variant, indicated it was de novo, although I will say that parental identity was not confirmed. Not that there was any suspicion, but something to think about when you're looking at de novo testing. So the laboratory reported this back as a variant of unknown significance, but clinically it was then interpreted as pathogenic, and this family was given a diagnosis for this patient of OI type 1. However, as the child was being followed, the subsequent treating physician was not convinced that this variant was the answer and really didn't feel like they felt the profile of OI. And so the patient and his mother were sent for whole exome sequencing. So what we found, interestingly, on whole exome sequencing is that the patient and his mother both carried a heterozygous 14 base pair deletion in the PLS3 or plastin-3 gene that results in a frame shift and a premature stop. And this is a relatively new gene, but it's a differential of osteogenesis imperfecta, and it's associated with osteoporosis, fractures developed in childhood, and hypermobility. Importantly, and a change from a de novo disease is that in this disease, which is X-linked, carrier women can range from normal bone density to early onset osteoporosis and fractures. So this is an example of our warehouse page where it's giving us a little more information about that variant, and this is what Runes is spitting out. So at the time, this was saying that we had seen this in only 2 samples, which was the patient and his mother. It's X-linked, and our patient was male, so that's why 1 was homozygous. The gene, again, is plastin-3, and then it gives us the transcript and how it's affecting the gene. Here, this is a program called IGB, which is put out by the Broad, and this is what allows us to visualize. So again, you can see that the patient on top is homozygous for the deletion, that black bar in the middle of the reads, and the mom is heterozygous for the deletion. This is just a screenshot showing some of the features associated with plastin-3 and how well it fits and overlaps with this family. So it's X-linked dominant. They can have osteopenia, osteoporosis, multiple fractures after trauma, vertebral compression fractures, and long bone fractures after minor trauma. So after doing the exome sequencing and talking with clinicians, this family was given, I think, the correct diagnosis now, molecular diagnosis of PLS3-related osteoporosis. Now, the mother is a carrier of this, and again, remember, mothers can be symptomatic, and so for her, we've recommended early screening for osteoporosis. He does have a sister that's not had any fractures, but we'll offer testing to her. She's been offered testing to determine her genotype because, again, females can have some manifestations of disease. And it also results in a pretty big recurrence risk change in this family. So before, what was thought to be de novo dominant, so we would have told the parents they had very little chance for recurrence, and we told the patient that he had a 50% chance at the time he decided to have children if he did. We've now changed in that it's X-linked, and so our patient has a chance of having only carrier females and unaffected sons, but now we need to talk to the whole family because there's other members at risk for having either osteopenia or having affected children. And in this case, the family was actually very happy with the change in recurrence risk because it decreased the risk in the proband, even though it increased risk for other family members. I think more than anything, this is really highlighting how before with the traditional medicine, when we were doing Sanger sequencing and gene by gene, you only found what you were looking for. So we only looked in collagen 1A1 and 1A2 because that's what we knew to look for, and we found something. But when we opened up and looked beyond the streetlights, so we looked in the areas that were previously dark, we saw, oh, this is actually a much better diagnosis or a much better fit. And we've seen that quite often in our family and in patients that have been sent where the initial diagnosis just doesn't quite make sense, or it's a variant of unknown significance that we're trying to get some more information on. I think this is particularly applicable, and this is one of the reasons why our center is within a pediatric hospital, is that in pediatrics, when you're early in disease progression, so when those children are first presenting to clinic, they often have incomplete symptoms. They're early in development, and so you can have a stereotyped adaptation to challenges. So if it's a metabolic disorder, if you haven't accumulated those damaging metabolites very long, you won't have the same disease as someone who's had the disease for 8 years. Or maybe they shouldn't be talking, so you don't know that they won't be able to talk. We're increasingly recognizing genetic heterogeneity, so several genes are causing the same phenotypic disease, where before we thought there was one gene, one disease. We're also seeing increasingly that there's very poorly defined clinical heterogeneity. There's only 2 people in the world that have been described with the disease. I think it's fair to say with quite a bit of confidence that we don't have a full understanding of all the symptoms that could possibly be associated with that disease or that gene. There's phenocopies, but I think another thing that most people can agree on is that an early diagnosis will give us the best chance for an optimal outcome. So the earlier we understand the disease, we understand the gene and the pathways, that gives us the best chance of finding a treatment or working and getting the proper interventions for that child. And at a minimum, that would give the families, or can empower the families in looking at family planning. So I also wanted to briefly talk on genome sequencing, because I think it really, more than anything, in addition to exomes, but it's highlighting how research and clinical are working together. And so as I mentioned, we do offer clinical whole genome testing, and this requires consent by a genetic counselor to discuss incidental finding options. So the American College of Medical Genetics has put out a list of 56 genes that they recommend be offered to all families undergoing full exome sequencing or genome sequencing. And these are primarily cardiomyopathy genes or cancer genes, but they have the option of getting results if there's a known pathogenic change that's been seen in those that we could report back to them. As a center, we've been offering this for almost a year, about 10 months. We run genomes on the child and exome on the parents, and in only 10 months within only our center, we've done clinical sequencing on 52 of our bands, which I think is pretty amazing because we go through a pre-certification process with insurance. But I think if we're looking at our testing and our clinical and our research and our genome, is that they're actually not that different after all. So in our clinical practice, we want to diagnose patients, we want to target our treatment, we want to avoid unnecessary treatments and tests, and we want to educate patients and offer genetic counseling. On the research side, we want to do all of these things too, but we also want to discover genes and phenotypes, we want to develop drugs, treatments, we want to innovate, so we want to look at new test methods, and we also want to illuminate disease state pathophysiology. But I don't think you can really have that research component now in the era of genome sequencing when you have clinical sequencing. Because what we can see and what does happen all the time is that you will see changes in a gene that maybe the gene hasn't been annotated before or been reported in humans, but there's certainly evidence to make it suspicious that this could be the disease phenotype. So we need a way, and we're all working on ways, to better do those functional studies and get the data so that we can report that back clinically. So one of the components too for our research in genome is that we're looking at the application of rapid genome sequencing in the NICU. And so this is that STAT-seq project. And this is where we can routinely do a genome in 50 hours under our research protocol. And so again, in the NICU, when we have critically ill children that are presented, this can be effective to diagnose or rule out known mutations, because both are important. And the idea is that if we can do that at the start, we'll reduce unnecessary tests. It can be useful and other testing, and again, we're going to cast that wide net to find it. And really, we were the first one in the world to do these emergency genomes. The result of this work is that we're a key site in a U19 grant that offers enrollment of up to 800 babies, and we're randomizing that to experimental control, where half are going to get the rapid genome and half are getting standard of care, which is going to allow us to see the clinical utility again of what if we offered clinical genomes in a rapid state and under a rapid basis in our NICU. It also is including some psychosocial assessment and 1-year follow-up. So again, a case speaks 1,000 words. So case 3 was one of our STAT-seq cases, and this was a baby that had a history of hypoglycemia with a glucose level of 18. They were transferred at 1 month of age to our center, and again, we were still having difficulty managing their glucose. They were the 4th child born to 27-year-old parents with one with similar concerns and really no contributory family history. They also had a normal newborn screen. And at the time, because we were unable to control the glucose, they were considering a total pancreatectomy. So as we're looking at clinically how they're working with managing these, they consulted CHOP for surgery for that pancreatectomy or partial pancreatectomy. And they said at the time, well, you're never going to have your genome results back in time for us to do the surgery because it was already scheduled, which was like the gauntlet had been thrown for us, so challenge accepted. So we consented them on the 24th, received the samples not until the 25th. We had a preliminary result, though, back by the 28th. We ordered our Sanger confirmation. Because it was under research, we had to clinically confirm before we could put it back. And we had the final clinical report signed and given back to them before they went to have their appropriate surgery at CHOP. And so what we found in this family is that they had a heterozygous known pathogenic variant in ABCC8 that was inherited from dad, but we didn't see a second variant. And this is classically related to autosomal recessive disease, except for when it's not. And so paternal inheritance is highly suspicious for focal disease, meaning that there was a second somatic event that leads to loss of the maternal allele in a subset of cells. So, but this results in a drastically different treatment and outcome. And so instead of that total pancreatectomy, which would have led to lifelong diabetes, the patient had a subtotal pancreatectomy, and essentially they're cured. When we looked at our first 45 affected individuals, which was 41 families, we had at the time of this study, when we did it, we had 13 families with a confirmed diagnosis. We had 3 additional that had a partial diagnosis, so that family probably had more than one thing going on. 8 families had candidate genes that needed further work before we could report them back. And we did still have 14 families with no diagnosis or candidate gene. And 8 of these diagnoses directly impacted clinical management. And sometimes that impact on clinical management, especially in the NICU, is a redirection to palliative care. And that's just as important as understanding that there might be a treatment. And so I think as we look at genomic medicine and what we're doing already, kind of to summarize what I've shown you, is that already we're able to use genomic medicine to rapidly diagnose our critically ill patients and to end those diagnostic odysseys. We're able to identify those treatable conditions earlier, and that's enabling us to find a quicker path home for patients. We're able to provide accurate prognostic information and recurrence risk for our patients, which will enable new or alternative treatment options. So we look at N-of-1 clinical trials. And of course, we're always discovering new disease genes that we're reporting. And so I think genomic medicine is already changing how we're managing our patients, and it's just going to continue to change how we're doing that. I hope I've shown you that through this presentation today. So I'd also like to just take a second to acknowledge our whole teams. Again, as I mentioned, it takes an entire village to run a genome center. And then there's a link to our website if you want more information about our center. So I thank you very much for attending the webinar, and I'd be happy to answer questions now.
Thank you, Dr. Farrow, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Farrow will answer as many questions as time permits, and the first question is, in a 2013 case study, you linked a 2 base pair de novo deletion to substitution of a valine for a glutamine and microcephaly, autism, global developmental delay, and metopic craniosynostosis. Are base pair deletions also linked from the Zika virus to morphological and behavioral phenotypes by amino acid substitutions?
Not super familiar with the Zika viruses, sorry, let me start over. I'm actually not familiar with the Zika virus and how it may or may not be interacting. It's probably not through base pair deletions, although viruses are known to be able to mutate themselves pretty rapidly. That's how they get resistance to different treatments.
As differential splicing results can take some time, how can NGS use show both splice-specific and environmental issues in pediatric diseases?
Splicing, that's a great question. And what I didn't touch on or go over is something called RNA-seq. So what RNA-seq enables us to do is actually sequence the RNA or the message RNA. And so we can use that. That's actually a very powerful tool for assessing novel transcripts. So if we have a splicing change that we think is changing whether an exon is spliced in or out, you can actually use RNA-seq, and that allows us to detect those alternative transcripts. One of the limits of that is that it has to be a gene where it's expressed in a tissue that we're able to get to. So we can easily get blood from our patients, but many genes, especially if we look at neurodevelopmental, aren't expressed in the blood. So that's one of the limitations. But as we look at trying to functionally characterize, especially our splicing variants, that's the first thing that we do is we look to see, can we get RNA from an appropriate tissue? And then that allows us to look at the splicing.
Did you prove functionally that these variants you found are the real causatives of the disease?
So for our analysis, what we do is that we follow the American College of Medical Genetics has also put out draft guidelines for the interpretation of pathogenicity and variants, and we follow those guidelines for our clinical reporting back. So in some cases, if someone else has done the work or it's very clear, for example, some genes are known or characterized by de novo base pair changes in the gene, then we don't need to do additional functional studies because the background of knowledge is already there. In the cases where we have either a novel mutation in a known gene or we have a novel mutation in a novel gene, then yes, we do undertake those functional studies either ourselves or through collaborations to help prove and give ourselves the body of data that we need to then give that back and report it to the patient. So that's where you'll see some case reports that come out or collaborations. So it takes a mix of both.
To what extent could we depend on the genomic data, especially if they are novel, to take a clinical decision specifically in cases that need prompt interventions?
So I think the question is, how are we using the data to guide our clinical, or what steps are taken? Again, we're relying on the ACMG before we report that back. If someone's in a research study, then of course they need to follow all clinical care as they standardly were, or would in case if the research study didn't exist. When we find novel changes in our clinical studies or new changes that weren't expected, things that we often do is that we have case conferences, or if we have calls or meetings, depending on where people are located, to really discuss and work through with the physicians. Like, this is what we see on our side. This is what, the molecular genetics is telling us. What do you see in the clinic? And we can work through that together to see, are there other things that we should be looking for? Are there other tests that we should be doing to confirm that? And so neither the clinic nor the lab is operating a silo in our institution. I think that's one of our strengths, is that we work together.
Do you teach students to differentiate between mutations and amino acid substitutions?
Sure. So we actually use those terms often interchangeably. So a mutation or a variant just means any change that we see in the genome that's different from the reference. So that could be anything. What can sometimes happen is that in our code, so in that central dogma of DNA, RNA, protein, when we're coding, or the amino acid, our 3 codons, DNA is coding for a specific amino acid, what can happen is that sometimes the change and the DNA can cause a change in that amino acid. So you may go from an alanine to a valine. Sometimes what can happen is that you have a silent change and that you have a nucleotide change, but it's not changing an amino acid. So all variants are mutations, but not all mutations result in an amino acid change.
What of your 2 NGS instruments has better QA/QS in terms of quicker treatments and cheaper cost?
So if I understand the question correctly, you're asking what are some quality things or measures that we put in place and ways to make it cheaper and quicker to move into the clinic. Some of those changes that come about are outside of our control. So a new instrument such as the 4000 is able to produce more data in less time. So that enables us to put more people together or more samples together on a single run. We have pretty stringent quality control metrics on our side that we use universally, and so those, if anything, are going to slow us down, but I think they're pretty critically important to make sure that we have the right data coming through. The other things that we're looking at is we're always looking at ways that we can use either automation to help us with sample preparation. Are there ways that we can change the sample preparation methods ourselves to make them go faster? And so again, we're using a combination of different factors to cut our times down. The other thing that there's seen a pretty dramatic change in, so not only in the sequencing time, but in the analysis, the compute time. So where it used to take us an entire day for a genome to be annotated, we can now do in under an hour if we need to. And so that's another place that you don't often think about a big decrease in time, but that us as well as others are working on getting that informatics side down and faster as well.
Did you have a case where sequencing information was not helpful or led to confusion?
Of course, we definitely have cases where we either don't have the diagnosis, and so particularly if we've run a whole genome, that can be very difficult for someone to understand that, well, you looked at everything and you're telling it must not be genetic, which isn't the case. It just means that we don't know where we just aren't able to find it. Or sometimes what can happen too is that we'll see something that we, on the lab side, say, wow, this seems really suspicious to us, and then on the clinic side, they'll say, that just doesn't make sense with what I'm seeing. And so, again, that's the strength of us being able to go back and forth. But we have some variants that we're just not sure. And so they get bucketed into that variant of unknown significance. And they kind of sit there until we either have more patients or we have a publication or, you know, we start seeing it in someone else. We're like, gosh, these people don't match up at all. It must not be it.
I would like to once again thank Dr. Farrow for her presentation. Do you have any final comments for us today?
No, I just want to thank everyone for listening. I hope that I've shown you the power of next generation sequencing and how it can be applied in clinic, and I hope that I've excited you as much as we are about how we're using it to help our population, and looking forward to all the changes that are going to come in the future.
Thanks again. I'd also like to thank the audience for joining us today and for their interesting questions. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, and to get your credits, click on the button located in the bottom left-hand corner of your web page. This webcast can be viewed on demand through November 11th, 2016. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.